Brain Science Institute. And I want to first of all recognize Kendall Renley, who helped put this film together and really put a lot of effort in a short period of time into it. Kendall, thanks very much. So today we are undertaking an ambitious and a slightly risky adventure. This is going to be spontaneous, it is not going to be scripted. And I want to pass along at the very start 2 sobering pieces of information. One is that there will be neuroscientists here. Now, a few years ago, my vice chair convinced me that we should give our neurology department the Myers-Briggs personality inventory. And so a total of 140 people took the test, and we had Hopkins corporate psychologists come to tell us what it meant. And he said, well, I've never seen anything like it. All of you are clustered down in one corner of being overly intellectual, judgmental, and earnest, and a variety of things that didn't sound too attractive. And he said, I don't know what to tell you. He said, I'm sure you get along well with each other, but I wouldn't want to go to a party with you. Well, I'm sorry to say that you're at a party with us. And so our goal is to loosen up, to have a sense of play and adventure that is a little bit foreign to us. And I think that is the challenge. This day should be fun, and along the way, we should laugh as well as think and inquire and adventure. The second piece of bad news is that we won't give you answers. We will give you questions. And so if you're here to find out how the brain does these things, you'll be a little bit disappointed. One of my favorite cartoons many years ago, when a man named Jerry Brown was running for governor in California. Well, he's actually running again now, but this was in the early 1970s, and he at the time was very much influenced by the ideas of E.F. Schumacher, that small is beautiful, that government should be much, much more restricted, and that people should lower their ideas of what government could do. Wonderful cartoon appeared that consisted of 2 panels. One was a road sign along a very empty road in the California desert with a sign saying, entering California, reduce expectations. And the second panel was a similar road sign with a sign reading, leaving California, resume expectations. So I would suggest that today, keep your expectations realistic, and we should have a good time. I want to point out in particular that Professor Samir Zeki from the Institute of Neuroaesthetics at University College London has come to be with us today, and we're going to use him literally in a very different way. We're going to ask him to volunteer comments or to respond by being called on as the day goes on, and I think we'll all benefit from his thinking and his experience. Now, this is the object that we're going to be talking about, and this will be a display that will be available, be able to be used by all the speakers. I would suggest to you that it is in itself not an object of neuroaesthetic beauty. It is a very clunky, very bulgy system. I want to tell you a couple of basic things about the organization of the brain. The first is that it's divided into right and left halves. Those halves in general work in a very coordinated way. A few of the functions of the brain are highly lateralized. Language is a good example. Spatial perception has a heavy lateralization to the right side that is often overly underlined. In 1980, a book came out called Drawing on the Right Side of the Brain, which had an enormous impact. It led to the idea that there was a right brain series of tasks, a left brain series of tasks, and in fact there were left and right brain personalities. There is now on the internet a 20-question survey that can tell you whether you're a right brain or a left brain person. In fact, there's a 1-question survey that is available, and that question is, do you spend more than 1 hour a week on the telephone? And if the answer is yes, then you're said to be a left brain person. Well, that is an overly reductionist view, to say the least, and we'll spend a lot of time talking about the fact that the brain works in very, very coordinated fashions. The second point, and this will come up in most of the sessions, is that there are very specific areas that do have special involvement in specific tasks. For example, the hippocampus that we will be seeing in a second is an area that is very much involved in memory acquisition and particularly, as we'll hear, in special aspects of spatial perception and spatial learning. And if we can see the area of the fusiform gyrus, this is an area that is very much involved in recognition of things such as faces. Other things as well, and it's not a specific role, but it is very much involved in facial recognition. As we talk about art, I'm going to tell you a personal story that really brought home to me the importance of this facial recognition area and how much we're wired to read faces. I developed a visual defect a few years ago that consisted of the inability to see what was just to the left of midline in a relatively small area. And that produced virtually no problem whatsoever. The world looked as full and as rich as it does normally, except when viewing someone's face where you didn't see the face whole. And that was a very distracting, distressing experience. I went to an art gallery where an exhibit of portraits was present, and I had to walk out because we can't integrate faces with that kind of defect. And this issue of special impetus or special importance on particular things that we see or that we hear or that we taste or smell, I think is one of the issues we're going to be talking about. I'll also make the point that what comes into our brains is not necessarily the seamless, full, rich world that we see or hear. It comes in a highly pixelated fashion, and we make it what it appears to be, and that will be one of the underlying topics throughout this session as well. Now, I want to conclude by introducing John Hamilton, who is, I would say, the most courageous person on the planet. John is at the science desk at National Public Radio. He's been very much involved in the conversations that have led to putting this program together. He is going to moderate a very spontaneous undertaking, and it could fail. This could be known as the time when John Hamilton's program died on stage. So, John, we thank you so much for being willing to take this on, and we really look forward to what's to come. John, thank you.
All right, with that introduction, let's get started. I am John Hamilton. I am a reporter on the science desk at National Public Radio. I'm going to tell you just a little bit about me because I think it may add a little bit of context to what's going on here. I am in no way a scientist. I've been asked probably 5 times in the last 24 hours since I've got here whether I am a scientist. I am not. I was an English major, and all that connotes. And I am also not an artist, of course. However, I should point out that many years ago, when I was at Oberlin College, I was at a place that was filled with both scientists and artists. And what I remember about that time was how many of the artists I knew were taking science courses, how many of the scientists I knew played an instrument or were in some way involved in the arts. So I guess I'm hoping that this event is going to take me back to my college years, and we'll just see how it goes. So on to the first session. It's about vision, and it's going to have a fair amount of science in it, fair warning here. One reason for this is that we need to sketch out some of the scientific concepts you'll be hearing about later on. So you might want to think about this first session as sort of a mural, a big backdrop, a landscape for the rest of the meeting. And let's go ahead and call up our first panel. Dr. Jeremy Nathans is going to be our first speaker. He's an MD and a PhD. He's also a professor at Johns Hopkins who somehow manages to work simultaneously in the departments of molecular biology, genetics, neuroscience, and ophthalmology. Not quite sure how he does that. But most important for this session, Jeremy spent a lot of time studying the biological systems that allow us to see and to make sense of what we see. So, Jeremy.
It's a pleasure to be here, and I want to kick off the session with a very brief overview of the visual system and in particular color vision. And let's start with A few basics about the brain. Here we see it in a magnetic resonance image. And I want to give you a few numbers to go with this. The human brain has about 10 to the 11 neurons, nerve cells. So this is a 1 followed by 11 zeros, 100 billion. The average nerve cell has 10,000 synapses, the points of communication with other cells. So that's 10 to the 15. That's 1 followed by 15 zeros, connections. How big is that? Well, if each connection were represented by the thickness of a piece of paper, and we were to stack 10 to the 15 pieces of paper, one on top of the next, the height of that stack could go around the globe 1,000 times. So that's a big number. And the total length of all the axons, the long-distance connections within a single human brain, if laid end to end, would go around the globe 5 times. So the brain is enormously complex. How could it be so complicated? It must be composed of pieces that are very small. So the brain is also very much miniaturized in terms of its component pieces. Here's just an image showing the connections between cells. Details aren't that important, but here's one cell talking to another cell and so on. And the pieces are about 50 nanometers in size. Now that's about the size of the very latest integrated circuit components. But let me just note before we imagine that the computer will match the brain anytime soon, integrated circuits are made in 2 dimensions, and the brain is 3-dimensional, and the brain is vastly more complex than today's computers. This is all by way of excuse to say that we don't understand how it works. Now, to vision. The visual system is, as you well know, one that integrates central nervous system components in different places. There is a part on the back of the eyeball called the retina, essentially the film in a camera, if you're thinking by analogy, which then sends information along the optic nerves to the brain's central region, which then sends it massively to the back of the brain. And Marge Livingston is going to have more to say about that. Let me just show the retina, if only because it's a beautiful picture, in a little more detail. This is actually a picture from 1887, quite accurate. And the main point I want to make is that there are layers of cells. So these objects that we're seeing are cells. These are the photoreceptor cells, the primary sensors of light. And these down here are the circuitry that analyzes that information before sending it to the brain. There are cones, small shaped sensors that mediate color vision, and then there are rods, the longer ones that mediate dim light vision. Now, just one word about physics before we go into color vision. The electromagnetic spectrum, that is the range of electromagnetic radiation in the universe, is very large. It goes from highly energetic radiation, gamma rays, X-rays, ultraviolet. A tiny sliver in the middle, the visible region, is the part we can see, and then infrared, microwave, radio waves, and so on. So we are capable of sensing just the tiniest little sliver of this vast array of information that's out there. Just parenthetically, I should say the astronomers are under no such limitations. They have telescopes, equipment that can see the entire range, so they can probe the universe with the entire electromagnetic spectrum. But within the region that we can see, let's discuss exactly what we mean by color vision and how it's used in nature. Now, obviously, color vision exists, that is, the ability to discriminate different wavelengths of light exists for one purpose only. It's not because it's beautiful, although it is. It's because it's information rich. That is, you can extract information from the world if you see it like this instead of like this. Information about the reflectances of objects. That is, if light shines on an object, say a red apple or a green apple, which wavelengths of that light will bounce off of the object and be seen by us versus which ones are absorbed. That's the thing we want to know about the world. And nature uses that to the hilt. So just a few examples. Flowers are differently colored, and they've been differently colored since well before we walked the planet. And they're that way because they are attracting pollinators, bees, butterflies, hummingbirds, and so on, who want to know which flower is which. Fruit is differentially colored, especially to indicate its degree of ripeness, again, because animals are eating it, and that's good for spreading the pits around. Animals are differentially colored in many cases to identify species. If you're a butterfly, it's hard to tell which species your future mate is from unless he or she is colored. Some animals have differential colors for the different genders. So, for example, males, in the case of cardinals, are highly colored, females are not. And other animals are talking to potential predators, telling them, in this case, for this poisonous frog, don't eat me, I'm poisonous. So there are many conversations going on in the world, in the natural world, using color as the currency. Now, this of course implies that color vision, the ability to sense those colors, exists in the animal world. And that then brings us to the question of how do we know if an animal has color vision, and what sort of color vision does it have? And the ultimate answer is we have to test the animal. And here's an example of a test. This little lady is looking at this set of 3 panels, and she has to decide which one looks different. It's this one, obviously. And if she pushes the button that says this is the one that's different, she'll get a little candy here. So we can test color vision in animals. And those tests, as well as various electrophysiological tests that don't require behavior, have indicated that color vision is very widespread in the animal world. We primates are quite good at it, but actually we're not the best. There are many animals that have more sophisticated color vision than we do. Honeybees, for example, many tropical fish, various birds, and so on. And each has a kind that is dedicated to its niche. Now, let's just focus at this point on human color vision. And I want to give a somewhat historical overview, partly because it's an interesting history. And the story begins in 1665, when Isaac Newton was a 22-year-old undergraduate at Cambridge University. Newton grew up in the English countryside. He was very familiar with rainbows. Rains a lot there. And he was interested in recreating the phenomenon of the rainbow in a more controlled setting. And so he drilled a hole in the wooden shutter of his dormitory room to admit a thin pencil of sunlight, and then passed that through a prism, and it split up into its component rays, red the least refrangible, that is the least bent, and violet the most bent. This looks like that Pink Floyd album, or I'm dating myself here. And then Newton did something very important with this. This had been appreciated before Newton. He realized that this was a method for purifying different colored lights in a way that was completely objective, that is based on their angle of refraction, not based on what they look like, but based on how much they bend when you pass them through glass. So he could put a stopper above and below, for example, this orange light, and allow that single beam to pass through. I can't resist showing you his original pencil sketch. This is Newton's sketch. Here's the dormitory window. It's left, right, reverse from the last one. Here's the light coming through. Here's the prism, and here he's allowing one beam of light to go through here, and so on. And the critical experiment, from the point of view of today's discussion, is this one. When he recombined the purified lights, that is, shining them together again on a screen, for example, a pure green, a spectrally pure green, and a spectrally pure red, the result is the appearance of yellow. Is that yellow identical, physically identical, to the spectrally pure yellow that has an angle of refraction intermediate between red and green? This is the most profound experiment in all of color vision. Newton proved that the answer to that question is no. It is not physically identical. It is identical in appearance, but it is not physically the same. How did he know? Because he took this compound yellow and passed it through yet another prism. And when he did that, it split up into its original red and green lights, each with the original angle of refraction. So the conclusion is that the human visual system is, in a sense, limited. It cannot distinguish certain physically distinct combinations of stimuli. I can't resist showing you a couple of things that Newton had to say about this, not that experiment in particular, but just in general. This is a century before the wave nature of light was appreciated. And here's Newton's speculation. He says, may not the harmony and discord, that is, the similarities and differences of colors, arise from the proportions of the vibrations propagated through the fibers of the optic nerves into the brain as the harmony and discord of sounds arise from the proportions of the vibrations of the air. So at this point, it was appreciated that sound consisted, partly from Newton's work, as a pressure wave of compression and rarefaction. And by analogy, he's suggesting that light might also have wave properties and have the same sort of aspect. I also can't resist just noting that this was an especially fertile time in the life of Isaac Newton. Here's his reminisce of what he did that year. He says, in November 1665, I had the direct method of fluxions. That's differential calculus he invented at that point. He's at age 22 now. At the next year in January, had the theory of colors. And in May, I had entrance to the inverse method of fluxions. That's integral calculus. And in the same year, I began thinking of gravity extending to the moon. And from Kepler's rule, I deduced that the forces which keep the planets in their orbits must be reciprocally as the squares of the distances from the centers about which they revolve. That was a very good year. Okay, onward. A century later, Thomas Young, a universal genius, gave the Bakerian lecture to the Royal Society, and in an almost an offhand comment, had this to say about color vision. Here's his speculation about the receptors in the retina that mediate color vision. He says, as it is almost impossible to conceive each sensitive point of the retina to contain an infinite number of particles, each capable of vibrating in perfect unison with every possible undulation. That is, he had just shown that light had wave properties. So each receptor would be tuned to one particular discrete wavelength. That's the vibrating in perfect unison. It becomes necessary to suppose the number limited, for instance, to the three principal colors, yellow, red, and blue, and that each particle is capable of being put in motion more or less forcibly by undulations differing less or more from perfect unison. That is, he's saying that each receptor has a point that it is most sensitive to, the point of perfect unison, but it's also somewhat sensitive to wavelengths that are somewhat more different from that. That is a sort of a broad bell-shaped curve of sensitivity, and there would be three overlapping receptors of just that sort, and this is exactly right. And now, 2 centuries later, we know those absorbance curves. Here they are. We have a shorter wave sensor. This is wavelength on the horizontal axis, a medium wave sensor, and a longer wave sensor. And all of human color vision is mediated by just these three types of sensors. Now, this has given rise to a very simple way of looking at color experience, and that is that it can be conceptualized as a three-dimensional space. Here in one dimension, the extent to which the long wave sensor is activated. In another, the extent to which the medium wave sensor is activated. And the third dimension, the short wave sensitive activator. So the color world is a three-dimensional world in an information sense. And if we take a slice through that three-dimensional world, we can recognize the arrangement of colors. Here's white in the middle, stimulating all three equally. And here's red, green, and blue. Now at this point, let me just note that there's an interesting complexity in terms of the human population. Not everyone has the same kind of color vision. About 8% of males and about 1% of females have variant color vision. Perhaps the most famous person to have that is John Dalton, the chemist, the 18th century chemist, who in his first publication, which is really a set of introspective observations, had this to say about his vision. He says, that part of the image, which others call red, appears to me little more than a shade or defect of light. After that, the orange, yellow, and green seem one color, making what I should call different shades of yellow. This is from 1794. So he was a dichromat. He was down from 3 receptors down to 2. He was missing, as we now know, his green receptor, his medium wavelength receptor. And what the world looks like, or looked like to John Dalton, if this is a trichromatic scene of a vegetable stand, or a fruit stand, this is what one type of colorblind individual would see. This is another. This happens to be the John Dalton sort, and this is a third. Okay, so I think you can appreciate this limits one's ability to extract information from the world. Now, for the last just few minutes, let me just say a few words beyond the primary receptors about what we do with that information. And it turns out that we do an awful lot. It's not simply transmitted electronic camera fashion, unprocessed to the brain. One of the first observations related to processing was from Ewald Hering, the great 19th century physiologist. Hering noted that there was something sort of unphysical, something counterintuitive about how we look at the spectrum of colors. If you think like a physicist, we could start at the long wave end, the reds, and we go, now this is the wavelength of light, we go to shorter and shorter and shorter and shorter and shorter wavelengths down to the violet end, and we've gone from 700 nanometers to, say, 400 nanometers. But the brain doesn't see it as a straight line continuum. It sees it as a circle. That is, the longest wavelengths, the far reds, seem to us to be very similar to the shorter ones, the violets. We connect it up as a circle. That doesn't make any physical sense, but that's how we do it. And so Hering suggested that although there are 3 receptors, they're actually organized into 2 color opponent processes. That is, red is opposed to green, and yellow is opposed to blue. And in that way, we get a circle in terms of our processing. And let me just demonstrate in a second for you that that is true. In fact, it is true. This conundrum vexed the field for a century, but it's now been largely solved. This happens because we do 2 things with visual inputs from the photoreceptors. We take the difference between medium wave and long wave sensors. That's one differencing operation. That's one channel. And in a second channel, a little more sophisticated, we sum the medium and long wave, and that gives us a sense of yellow, and that is subtracted from the short wave, the blue. So here's yellow minus blue, and here's red minus green. That's the information that goes to the brain. And let me just give you a nice example, a participatory example of one of the effects of this. If you will just fixate on this black spot in the center for 10 seconds, I'm going to show you afterwards, don't let your eyes wander from that spot, I'm going to show you a perfectly blank white slide. Okay, 5, 4, 3, 2, 1, presto. And you should see the complementary colors, right? What have we done? Let's go back 1 second. What we've done is, as you were staring at this black spot, for example, up here, every one of the photoreceptors in this region was receiving this blue light, and that limb of this differencing operation, it's like a seesaw, was getting fatigued. So that when we suddenly switch to white, the other limb, being stronger, is then enhanced. And we can just do it one more time. Stare at that little black spot. 5, 4, 3, 2, 1, poof. And each one has been reversed to its complement. Okay, now that's just the first step. The way that we see the world is highly dependent on interactions with not just the object of regard, but the surround as well. We are making judgments. We're analyzing the scene and reprocessing it based on the surround. And let me show you a very dramatic example. This is a picture of a cube, and I'm going to demonstrate what's called color constancy. That is the ability to judge a color based not just on the lights emanating from that object alone, or the wavelength composition from that object alone, but a sense of all the surrounding objects. So here, for example, the cube has been illuminated with a yellow light, and here it's been illuminated with a blue light. Note this blue square in the corner here, and next to it is a yellow one, and here's the blue square, and there's the yellow one. And now I'm going to tell you something that you won't believe. The light that is coming off of the top of this blue square right here in the corner is identical to the light that is coming off of this yellow square here. It is exactly the same photons. You don't think they look the same, but they are the same. And let me demonstrate that in 2 ways. We're going to crop it by separating it from its surround, separating these 2 objects. And the more we separate it, the more I think you can appreciate this is identical to that. This is not a Photoshop trick. This is real. But when they're integrated with the surround, they look very different. Let's just do that in a different way. We're going to crop the image and enlarge it. Here's just a totally different way to do this. We enlarge it a little bit. Now we're looking at this blue top and that yellow top. They look different here, but they're going to get less different as we enlarge it. This blue one, that yellow one. Blue, yellow, blue, yellow, blue. yellow. They're identical. You don't believe me, but to prove it, I've got handouts here. You can take them home, homework assignment, cut out from a piece of paper a little hole and put it over the blue one, another little hole, put it over the yellow one so you can't see anything else, and then look at what comes through those two little holes. They're identical. This is just the beginning, though. If we look at this effect in various other domains, for instance, lightness and darkness, we see the same sort of sensitivity to what's going on around it. This panel is identical to that panel. No difference whatsoever. It's just that the surround affects our judgment of their intensity. This white in the middle is identical to the white that's out here. But because of this gradient of brightness going from dark to bright, we judge the center to be much brighter than it actually is. And then there are more complex learned effects based on depth, for example. So here's a scene of a zigzag wall with two gentlemen walking. This line, this darkened line right here, is exactly the same size as this line right here. You believe that? Hold your hands up. Just do a little measure. Measure it. Just hold it up to the one line and move it over to the next one. They are exactly the same. That's on the handout too, just to prove it. Why? Why do we think they're the same? Because we've learned in the world that things that are far away, even if they subtend the same angle on the retina, are bigger. And we can't convince ourselves otherwise. And one more, finally. We also, and this again is a learned response, here's an image of cuneiform on the left, and it looks like the little bumps are coming out at us from this clay tablet. And here on the right, cuneiform also, but they're going into the tablet. Everyone agree with that? These are actually exactly the same photograph. I've simply taken this one and turned it 180 degrees. I rotated it around for the other one. It's the same image. The reason it looks like it's coming out on one side and going in on the other is we assume that the lighting is from above, without even thinking about it. We assume the sun is up, and therefore shadows are down, and therefore this is coming out, and this must be going in. So there's a combination of innate processing and also learned processing when we look at the world, and that gives us this richness, which you can't just predict by looking at the image alone, pixel by pixel. Thank you very much.
Reminded me of something my 4-year-old son said for a year before he figured out it was incorrect. He used to say, he would see something and he would say, "I can't believe my mind." And we used to correct him. We say, no, you mean you can't believe your eyes. Now I realize he was right the first time. Next up we have Marge Livingston. She's a professor of neurobiology at Harvard, and she's done some groundbreaking work on how the brain processes visual information, especially color. Marge also has not shied away from applying what she's learned to art. A few years ago, she got a lot of attention for explaining the Mona Lisa smile and why it is so elusive. Marge.
So I'm a visual neurophysiologist. I try to figure out how we see by recording from neurons in the visual system. But I acknowledge that artists also try to understand or make use of how we see. And sometimes artists figure out stuff that's so fundamental that we can understand it in terms of the underlying neurobiology. So one of the things that artists have known longer than us neuroscientists is that color and luminance can be treated independently in a work of art. And by luminance, I mean, for those of you in the art field, value. So Picasso said, colors are only symbols. Reality is to be found in luminance alone. The basis for this is that, as Jeremy said, we have 3 cone types. We calculate color by comparing the activity in two of the cone classes. We calculate luminance or value by simply summing the activity in the three cone types. So in this image, in this image, the color contrast, the color version is on the left, the luminance version is on the right, and most color contrasts are accompanied by a luminance or value contrast, but you can have color contrasts that have no luminance or value contrast. And very peculiar things happen at equal value or equal luminance, and artists know that. And I think some of them take advantage of that. And one of the first artists to really use this, although I'm not sure he did it on purpose, is Monet. This is Impression Sunrise, which is, in the original, the sun is absolutely brilliant. It vibrates when you look at it, and it looks really bright. But if you measure the value or luminance in Impression Sunrise, you discover that the sun, although it looks really bright, is actually exactly the same value as the background. And I think it's because it's equal value, equal luminance, that it has this peculiar quality. And that's because our visual system, the primate visual system in general, after primary visual cortex, is subdivided into these two major processing streams. And the ventral processing stream is sometimes called the what system, because if you get lesions in your ventral stream, you can't recognize objects. These are called the agnosias, so someone can show you something and you just can't tell what it is. You can draw it, but you couldn't tell what it is. You can lose the ability to recognize surprisingly specific categories of objects. So for example, people can lose the ability to recognize fruits and vegetables, or man-made objects, or faces, which means that there are particular parts of our temporal lobe that are dedicated to processing those particular categories of objects. The dorsal stream is evolutionarily older. It's what we share with other mammals. So the ventral part of our visual processing stream is a primate-specific add-on, and primates are better than other mammals by a lot at recognizing objects, especially objects with fine details, and certainly faces. Other mammals use their visual system to navigate in the environment, so the dorsal stream does all the things you'd expect a self-respecting visual system to do. It allows you to tell when something's moving against the background. It's sometimes called the where system because it seems to carry information about motion, three-dimensional organization, space. All of these are positional information, but it's also sometimes called the how system because you need this information in order to interact with your environment. So the where system or the how system carries information about motion, depth, space, and figure ground segregation. So here are some drawings made by people with lesions in their dorsal stream. These individuals were asked to draw a bicycle, a cross, and a clock. And you can see that these individuals got all the bits right, but what they got wrong was the spatial organization of the bits. And these look sort of like cubist drawings, right? This is not to say that Picasso had a parietal lobe lesion, but rather that he may have cottoned on to the idea that positional information and object part information are two different things. So now I want to talk about the ramifications of the fact that this evolutionarily old part of our visual system is, like other mammals, essentially colorblind. And I'm not talking about insects or birds. Other mammals do not see color the way primates see color. And so I want to try to convince you that your ability to see motion, depth, spatial organization, and figure ground segregation is carried out by a colorblind part of your visual system. So if you had a red spot on a green background and you took a black and white photograph of it, you'd get a gray spot on a gray background. And you can imagine that you could tailor the brightness of the red in such a way that the two grays would be identical. That's equal value or equal luminance. And weird things happen at equal value because there's a whole part of your visual system that can't see it. And you get peculiar effects at equal value, and it isn't because of the saturation of the color. So here's some gray spots And for me, this gray spot, and mostly that one, let's see the peculiar shimmery quality that you get at equal value. So I want to try to convince you first that your ability to see depth, spatial organization, and figure ground segregation are carried by a colorblind part of your visual system. Oh, first I have to define depth and spatial information. So the world looks vividly three-dimensional, right? Okay, I know it is. I'm not denying that. But vision is not like sonar. You don't send out a signal and ask how long it took to come back. How do you calculate depth and three-dimensionality if you're starting with two flat retinal images? It's a difficult computation. So your visual system, starting with these two flat images, uses things like relative motion, shading, perspective, occlusion, and stereopsis to calculate distance and depth. So first I want to try to convince you that your ability to see depth from motion is carried by a colorblind part of your visual system. And to do this, I want you to look at this thing, which I hope looks three-dimensional. Do we all agree? Come on. Need a little, okay? I hope it looks three-dimensional when the red spots are darker than the background or brighter than the background. Do we all agree? Don't worry about which way it's rotating. It's ambiguous. Which way it goes is postgraduate. We can talk about that later. But if I turn this, yeah. If I go through equal luminance, I hope this thing goes from being round to being flat and squishy and back to being round again. Does everybody see that? So that's your ability to see depth from relative motion, not doing so great at equal value. So in this image, I think you can tell that there's a lot of cues about shading, but I bet this thing doesn't look particularly three-dimensional until I eliminate the blue light and turn this into a luminance contrast image. And I hope this looks more three-dimensional when there's a luminance or value contrast than when there's no value contrast. And in this image, again, you know that there are cues to perspective, depth from perspective. There's figure ground segregation. There's all kind of stuff going on that's really hard to see in this equal luminance image. But if I introduce a luminance contrast, I hope this thing looks more three-dimensional and it makes more sense, right? You can't even figure out what's going on until you get rid of the blue. This is part of Liechtenstein's Rouen cathedrals, and I bet it doesn't even look like the Rouen cathedral until you introduce a luminance contrast. So if your ability to see depth from shading is carried by a colorblind part of your visual system, it shouldn't matter what the colors are in the shadows as long as the luminance or value is appropriate. So I think some artists played with this. This is André Duran's portrait of Matisse. If you knew Matisse, you would probably recognize him, even though the shadows are green, which is not an ordinary skin color. But although the shadows are peculiar colors, the luminance is appropriate to give you a sense of three-dimensionality. Matisse did this in Lady with a Hat. It offended a lot of people. But he's doing what Picasso said. The colors are symbolic. The reality is there in the luminance alone. And I hope Bill's going to talk about how he uses color, because he does the same thing in some of his paintings, which is the colors are peculiar, even more than shadows, his highlights are peculiar colors, but they're appropriate to convey a sense of three-dimensionality and light bouncing off this individual's face or eye. So now I want to try to convince you that your ability to see motion is also carried by a colorblind part of your visual system. And to do this, I'm going to show you some moving stripes. And the stripes are always moving at the same speed. You can cheat if you want and look at the center, but what I'd really like you to do is to look sort of to the right of the black and white stripes and think about the speed of the red and green stripes compared to the black and white stripes. And if I make the green stripes darker than the red, they still look like they're going the same speed, but if I creep up on equal value, I hope that you see the red and green stripes moving more slowly. Now they're fast, and now they slow down. Now they're fast again. So equal luminance stimuli are a suboptimal stimulus for your motion system. Artists may not be so interested in that, but I hope they'll be interested in the fact that if you can't tell that something is moving, you also can't tell that something is stationary without your dorsal stream. So I think a lot of artists, again, starting with Monet, made use of this to generate an illusory sense of motion in a static image. So Monet's poppies can be seen by your what system, but they're invisible to your where system, so their position can change. They can seem to move in the breeze. I don't know if Monet actually thought he was achieving a sense of motion, but I know Mondrian did because he named this painting Broadway Boogie Woogie. Obviously, he knew it was moving. It moves because the yellow checks are apparent to your what system, but invisible to your where system, so their position can seem to change. I don't like to look at this one, I don't have to. Advertisers do this all the time. You can't open Wired magazine without seeing some kind of text that's equal value with its background, and it has this jumpy, jittery, jazzy quality that's kind of unpleasant, and it's hard to read, but it gets your attention, which is what they want. So the ability to achieve a sense of motion in a static image is something that artists have been fiddling with for hundreds of years. This is Poussin's Rape of the Sabine Women. The longer you look at it, the more static it seems because nothing changes. Conversely, in Monet's paintings, the longer you look at them, the more you seem to get a sense of motion, especially in the water, where he has a bunch of equal value colors next to each other. So I hope I've convinced you that your ability to see motion, depth, spatial organization, and figure ground segregation are all carried by a colorblind part of your visual system. Now I want to talk about the fact that color is carried by a low resolution part of your visual system. Artists knew this before us neuroscientists. It's because the cells that carry information about color have big, low acuity receptive fields. You have smaller receptive fields for cells that are concerned with contour. So you do not have to color inside the lines. Watercolor artists, pastel artists, all use this technique of making a line and having a blob of color that doesn't stay inside the lines, and it doesn't matter because the color spreads until it hits a border. Your visual system doesn't use color per se to define the border. It lets the color spread till it hits the border because the color is low resolution. So I want to, here's an illusion based on this. I hope you see this part of the image as yellower than this part or this part. Do you? Come on. Okay. Okay, it's not. This was made in Photoshop. This white and this white and this white are all the same. This is a yellow line inside a blue line, and the yellow color spreads until it hits the next border. So now I want to use after images, and thank goodness Jeremy defined this for me. I want to use after images to create some blurry colors so that you can watch color spreading until it hits a border, okay? So what I want you to do is look at this intersection between these three shapes, okay? And then I'm going to give you an achromatic gray background where you'll see sort of an after image. Now do it again. Look at the intersection and evaluate the after image in this achromatic version. Is better the second time? Okay, now some of you are saying, well, you did it twice, so you built it up. No, that's not it. Look at the intersection, evaluate the after image. Look at the intersection, evaluate the after image. It's stronger when you've got a border. Now here's another variant on this. Look at the intersection and see if you care that one of the shapes changed. And now lastly, look at the intersection and watch the colors squirt out the hole. Another corollary of differences in acuity in our visual system was, I think, made use of by Leonardo da Vinci. This is the Mona Lisa, which is a very famous painting, and there's a reason why everybody loves this painting. The art historians say that her smile is ambiguous. Leonardo used sfumato. And that it can come and go depending on what you're thinking about it. But I noticed that, I was reading an art history book because my editor told me I had to read an art history book. And he said, notice how her expression changes. So I did, but if you look at her eyes, So look right at her eyes and think about her mouth. I know this is hard to do, but do it anyway, okay? Look at her eyes and think about her mouth. Now look directly at her mouth. Go back and forth between looking at her eyes and looking at her mouth. Bam. And see if you think she's smiling more when you look at her eyes than when you look at her mouth. So that's not ambiguous. No, cover it up. You have to look at her eyes and think about her mouth. You cover it up, it won't work. That's not ambiguity. That has something to do with where you're looking. And as a visual physiologist, it's like, I know that your acuity falls off from your center of gaze. It doesn't seem that way, but if you look at the central dot, all of those letters are equally readable. So your visual acuity is much higher in your center of gaze than it is in your peripheral vision. That's why you move your eyes when you read. But your peripheral vision is actually better at seeing big, blurry things. So the Mona Lisa, if you filter the Mona Lisa in such a way that you can see what she looks like to your peripheral vision, she's smiling from ear to ear, but if you can see the whole thing with your central vision, that's the panel on the left, she's hardly smiling at all. So as you move your eyes around, and you naturally move your eyes around constantly, what you see changes. And it also gives it a sort of a coy quality. So you're looking at her eyes, she seems to be smiling, you try to look really quick at her mouth, and she stops. But this gives a static painting a dynamic quality, which is something that 500 years ago was pretty special. You can see the same thing in photo mosaics. With your central vision, you see baseball cards. With your peripheral vision, you see Babe Ruth. So as you move your eyes around, different aspects of the image are what you process. Lastly, I want to talk about how your ventral stream processes objects. So we're really good at seeing faces. These faces are flashing by in about 100 milliseconds, but you're not only detecting faces, but you're recognizing individual people. And we can recognize faces, whether they're caricatures, whether they're degraded, whether they're occluded or disguised. And we can do this better than any computer program. And we do this with a particular part of our temporal lobe that is dedicated to processing faces. Primates, all primates do, well, monkeys, old world monkeys do this. And both macaques and humans have dedicated regions in their temporal lobe for processing faces in particular. And in my lab, we've recorded from cells in these regions. And we've asked, how do face cells distinguish one individual from another? So we showed some face cells, this movie, essentially, in which all these different face parameters are varying constantly. So we're varying eye size and eye position and mouth shape and face shape and all this stuff. And we're asking, does any individual cell just respond to one of those faces, or does it respond to particular parameters? And what we found was that individual cells care about one or two or three of these different variables, and only one or two or three, but different cells care about different sets of variables. And you might imagine that if you had a cell that cared about inner eye distance, that you'd want to have a cell for every potential inner eye distance in a plausible face. But that's not what we found. What we found was that there were cells that cared about really big inner eye distances, and then there were cells that responded best to really small ones. And this was true, so that's actually more efficient, right? Because you only need one of each cell to do it. And you get everything in between for free by looking at the ratio of the two cells. And so here's a typical face cell. These are all the different parameters that we looked at. And you can see that for each of the parameters that this particular cell cared about, the tuning is ramp-shaped. So that the cell responds best to one extreme, and you notice that those faces were pretty extreme, right? The cell responds best to one extreme and is inhibited at the other, or vice versa. So just like Jeremy said, you get a push-pull organization. You have cells that are in opponency, and what the cells care about is how each cell, each face, differs from the average. So faces are processed by how they differ from the average face, And I want to illustrate, so what I want you to do while I'm nattering on here is look at these numbers in the middle, okay? So what you're going to be doing is, like Jeremy showed you, you're going to be adapting. We're going to be creating an after image, and I'm having you move your eyes so that it's not a simple retinal after image, but it's a high-level individual identity after image. And now what I'm going to do is show you two identical morphs, but I bet you saw an identity after image, which is now fading away, right? These two images are identical. Artists know that that's how we process faces. Artists know that a veridical line drawing is more difficult to recognize than a caricature of an individual. So it's much easier to recognize these caricatures than it is to recognize the veridical line drawings, because we process faces by how they differ from the average. Picasso does this. You can see that these are typical Picasso portraits, but if you look at the photograph of the person these portraits are of, you see that they're actually a caricature. This is Picasso himself. This is his girlfriend, Igor Stravinsky, his art dealer, his mother-in-law. So vision is information processing, not image transmission. It doesn't do you any good to transmit a high-resolution image up to your visual cortex because there is nobody up there to look at an image. Vision is all about extracting from the environment information from the pattern of light on your retinas. And how one face differs from everybody else is information about that individual. You can see this in children's art. You ask a 3-year-old child, any 3-year-old child, to draw a person, and you will get a face on legs. As a child gets older, you'll get a body, but the body is not as important as the face because our faces carry social information and identity information, so they're the most important part of what we see in an individual. As a child gets older, you'll even get digits, but you'll get fingers, never toes. And if you ask a man, what are the, even 45,000 years ago, what are the important features of a woman? I think, and I hope Ed Connor is going to talk about this a little bit tomorrow, even the way we recognize objects may be similar to the way we recognize faces by figuring out how that object differs from the background and from other objects. So Matisse recognized this, again, long before us neuroscientists. He said, I don't paint things, I paint the differences between things. So vision is information processing, not image transmission, and artists often take advantage of the way of the algorithms our brain uses. They may do this empirically, they may do it explicitly, but they just use different words than us neuroscientists to describe what they're doing. Thank you.
Try not to kick that. All right. So now that we have learned a little bit about how easy it is to manipulate all of us, let us meet a professional manipulator, WLStoehr. He is an artist, a sort of unlikely artist. Until a few years ago, he still had a day job at National Geographic, not just any day job either, I should say. He was actually in charge of their worldwide mapping organizations, operations, which would raise us some questions about brain mapping, other types of mapping. These days, Bill is painting. It's amazing stuff, and I'm not going to try to describe any of it because I'm hoping he's going to show you some. Bill.
Can you hear me okay?
Excellent, good.
Well, Jeremy and Marge talked about the neuroscience of how we see and perceive color, line, shape, value. Marge showed us how artists foretold some of these same things. So I'm going to talk about how I discovered these same things. So it's a discussion on my methods and my experience in discovering and applying these visual processes. So to start things out, for me, the notion of aesthetics is really slippery, and I truly believe that anything can be art. But I've chosen a very traditional path, and that is painting canvas, figures, and faces. So I do big faces. And when I started painting 6 years ago, I was unwilling to start at the beginning with classes and lessons, and I just started to paint. I painted what I saw. And 6 years ago, you heard I left the best job I ever had at National Geographic, being in charge of their mapping group. So let me just talk for a moment about mapping. So here's the typical map, and you're all familiar with National Geographic maps. And today, I'm really using the same tools, the shape, line, color, to produce art. Back then, it was to produce two-dimensional maps. But so now I'm an artist using these tools, and there's a big difference. In mapping, it's very precise, it's accurate. There's no experimenting. There are no accidents. Accidents at National Geographic maps make the evening news. But as an artist, that's what I do. I experiment with visual perception. Accidents are a big part of what I do. Experimentation, I don't worry about accuracy, and I observe the results, and then I move on from there. So, keeping that in mind, I'm going to talk now about my process of discovery with big faces and eyes, caricature and line, selective detail, color contrast, and value or luminance. So a couple things about me. I'm color vision impaired. I now work with both my right and my left hand after having an accident. I never pre-plan a painting. I don't take notes along the way. I don't use an organized palette. I don't use measurements. I don't use formulas. I just paint what I see. So 6 years ago, when I started painting, someone told me, well, if you really want to learn how to paint, draw the human figure. And so that's what I did. But I've always preferred my drawings or my drawing-like paintings to my actual finished paintings. Finished paintings always became very tight for me. I just wasn't as interested in them. And my goal has been since then to paint like I draw. But no matter what I did, people kept saying the same thing. I love your faces. They were drawn to the faces, and they kept talking about it. And then a friend of mine said, why don't you focus on painting big faces? Then about the same time, I saw this painting. This is Marlene Dumas. And also about the same time, I found out that she had an exhibit in LA opening. And so at about the same time, I boogied out there, hopped on a plane and headed to LA. And I loved what she did. I loved her caricature-like faces. I loved the expressive eyes. I loved the red. And I loved the way she used the red. And I was already doing faces, eyes, and red. So this was an early painting of mine. And then I read that Mark Rothko said something to the effect that I paint big to be intimate, to encounter you. And I thought, isn't that exactly what Dumas was doing? She was painting big, and she was encountering me. And that's what I wanted to do. So I started painting big faces. And I put myself in these images. A few of them you'll see. So you'll see the size of them. And this is the first series of big faces. These are the small big faces. People had strong gut reactions to them. They reacted in ways that they couldn't, didn't, and wouldn't verbalize to me, but they just reacted. Now they're starting to talk about it. But at first, people really reacted strongly. And I was just painting local women, but at some level, viewers were identifying or projecting upon them. And I wondered, what is it that they're sharing with this image? Or what is it that they're telling me about themselves? Or what is it that they're sharing with me? And more than simply replicating visual appearances, I knew that I was tapping into something magical here. And it went beyond just simply faces and eyes and red and big. And I wonder today, what's the shorthand here that's involved? How does this visual perception trigger an aesthetic or an emotional response in some people? And I don't know. I saw Picasso's Guernica for the first time just a couple of weeks ago. I came up to the room, and I almost didn't go in. I couldn't go in. This canvas is packed with faces, caricature, with light and line and metaphor, I could hear the screaming. I could feel the heat. I could feel it on my skin, and I was afraid to go in there. Well, I did go in. Now, I'm not surprised by my reaction to this painting, to Guernica. But several years ago, I was in Amsterdam, and I came around a corner and was confronted by Van Gogh's Wheat Field with Crows, and I broke down and cried. I've never done that before. Now, it wasn't till years later that I discovered this was his last painting. This is not a painting of a horrific scene like Guernica. So how did it solicit such emotion to me? Well, I would like to find that out. So I'm interested in why people react to my work, because they do react to my work. And it's the direction I'm going. And I sense that this all starts with visual perception. And that perception is somehow making a connection somewhere in our mind that's triggering this rush of emotion. And that's what I want to know more about. So let me tell you how I work real quickly. I do a sketch from a live model. Again, this is a bigger painting. I do a sketch from a live model, and then I take a chunk of charcoal about the size of my fist, and I spread it all over the canvas. I take a wet sponge, and I move it around as if I'm finger painting. And it's just the way I started to paint. So that's how one would look at this point without the color. And then I add black marks, and then I typically drip, splatter, or pour on a series of colored glazes, real thin paint, and I give it just, a little bit of color. Then lastly, I get a brush full of red paint, and I very aggressively stroke on the red paint. I've put a hole through a canvas. On some occasions, I actually hand the brush to the model, and I say, here, you put it on. Well, I knew right away that people went straight to the eyes, because they talk about it all the time. They said they did. And from the very start, I've highly detailed, well, it's not highly detailed, but I put in a fair amount of detail into the eyes. And this selective detailing seems to increase people's interest in that. It seems that they like to go where there's some detail. And if I combine the detail in the eyes, well, then I've got something going for me. And then more often than not, the whitest white and the darkest dark are in those eyes. It's the pupil and the highlights. And so, again, I think that's attracting to people, and I guess I don't need a brain surgeon to know that people are going directly to that point, and they're telling me this is the focal point. And people seem to be wired to go to that. So let me talk a little bit about color. First of all, very early on, if the crayon was in the box, I used it. And I think that probably goes back to childhood, because I always got the small boxes. And when I was confronted with a lot of colors, this is a lot of fun. And so that's what I did. And that's a big change from where I am now. The other thing I do is I try sometimes to use the colors that I see when I have my eyes closed. Why do I see color when I have my eyes closed? And it's usually when I'm very relaxed and I get this rush of color. And so for a while I was trying to use that in my paintings. Early on, I was also very attracted to people that use color like Matisse, Bonnard, Wolf Kahn. This is a Wolf Kahn. And I looked at this painting and I said, how did he make those trees shimmer? So I'm guessing these are aspen trees, and it looks like they're doing what they're supposed to be doing, quaking. And how did he do that? But then I started to use some of his same color combinations in my own paintings, and they seemed to move, and they had some depth, and I got curious about that. And so one of the tools I like to use is Adobe Photoshop. And so I plug in photographs of paintings I take. I'll take it from a book, or I'll put in my own paintings, and I'll analyze the different color combinations that I might be able to use. So let's take a look at Wolf Kahn's shimmering trees here. Well, the first thing you know is that he's using complementary colors in here, isn't he? It's red and green, they're opposite on the color wheel. But if I turn it to grayscale, they're essentially the same value. So that's how he's making the trees shimmer. He's using, some people call it discordant color, but he's using complementary colors opposite on the color wheel, and they're essentially the same value. Everybody knows that. I didn't. So this was a discovery for me. So again, that's part of my process of discovery. Everyone knew that discordant colors did funny things, and I didn't. So here's some discordant colors. essentially opposite on the color wheel, and you see them in grayscale, and guess what? They're equal luminance, equal value. Well, I decided to try this with eyes. So I put a little green in the eyes, I put a little red. There was nothing to stop me from putting in yellow and purple, and pretty soon the whole color wheel with blue and orange, and the eyes started to dance and shimmer because I was using discordant colors. And people said that the eyes looked real. And I thought, that's great, but real? When was the last time you saw someone with purple and green eyes? It's not out there. But it didn't matter. I was putting red, green, purple, and yellow in the eyes, and they seemed more lifelike. But what I learned is the colors didn't have to be lifelike to produce a lifelike image. Okay, so now picture yourself in my drawing group, and we've got a model there for a 10-minute pose. And you get the basic figure down, okay? You're feeling pretty good about that. Then you start working in the eyes, and you get the eyes really good. And you like to do eyes, so you spend a lot of extra time on them. And then someone says, we've got 1 minute left. So you panic, you scramble, and you scribble in the rest of the face, and you look at it, and you say, wow, that looks pretty good. So I started to tell people that all I had to do was draw good eyes and suggest the rest of the face, and your mind would finish it for you. I told people that the worst thing I could do was to draw a highly detailed hand, not as an excuse, but I shouldn't draw hands are very hard to draw. No one likes to draw hands. But I would tell them I didn't need to draw those highly detailed hands if it detracted from the focal point. Why? Because people were drawn to the detail. Then I told them I didn't have to color in the right place. I just had to be close. Your mind would put it in the right place. I said it was okay to color outside of the lines. What's more, by doing that, it seemed to add rhythm and movement to the composition. And the colors, of course, didn't need to be lifelike to produce a lifelike image. Now, I may not have been completely accurate there, but I intuitively knew that the brain was doing some of the heavy lifting here. So if I engaged you with an affecting pair of eyes, I could create vague shapes, line and color, like this. And when placed in context, it seemed to move and tremble and be recognizable as something real. Okay, how else could I use this? I love Franz Kline. Well, he didn't do a lot of figurative work. Did some, but not a great deal. But I'm always trying to figure out how can I put these strong black lines into my paintings. And so I started playing around with it. So this is a Kline. This is another Kline. This is a Kline. This is mine. This is mine. And that's my painting. So I figured out that I could use these bold, black, Kline-like lines for other features, other body parts, and people didn't care because they were focusing on the eyes. And this allowed me to create much stronger compositions. Okay, let's talk about light. This is one of those quick, maybe 30-second sketches that artists do. And I created a life-size painting based on this sketch. Painting just wasn't coming together. It was a big painting, and I was painting out of doors. And just sitting over here to my left was the garden hose. So I thought, I've got this partially dried painting. I wonder what would happen if I just hosed the sucker down. So I picked up the hose, and I hosed it down, and this is what came out. And for the next year, I was trying to figure out, how did I create this illusion of light? And I looked at how other artists did it. I pulled out the books. What did Turner do? How did Rembrandt do? And I was a little dense about this. It wasn't coming through. I couldn't figure it out. And then on a different painting, I was doing what's become a signature of my style, which is to feather the edges of these big canvases of charcoal. And I started to see this gradual value change. And I noticed that it started to look like it was creating light. And I went back to this painting, and I finally recognized what I was doing. I had this juxtaposition of this abrupt contrast, right against this gradual value change. And that's just like Turner and Rembrandt, although I doubt that they were using garden hoses on their paintings. But it was the same idea, and it worked. And that's how I learned it. It was a pure accident, It was a playful experiment, and I learned something from it. So back in my studio, I frequently changed the order of paintings during the course of working on them. And I noticed that they worked as a group, and they worked very differently when I changed different paintings out. And it was both graphically or visually they changed, but they changed how people reacted to them and how I reacted to them. And that's when I understood that the room was my substrate or my canvas, but that's another talk. And by the way, that's how I display them now. They're up on the ceiling and they're down along the floor, and I put them together in these wild groups, but that wasn't enough. And I began to think about Rodin, and I thought, it's exactly what he does, what he did, didn't he, in so many cases. He repurposed and rearranged sculptural components for different effects. So I thought, well, how can I do this with my paintings? And this led to me to something that I refer to as proximity effect, which is something I came up with, but it's also about global abstract and local detail perception. So I thought, in a large gallery, how can I arrange faces like this and allow them to shift and change people's perceptions? And computers, of course. So I created this large-scale, maybe 12-foot video computer-driven exhibit, and it randomly selected faces, and it reprojected them every 3 seconds. And I could squint at them, and I could create this global abstract, and then I could back up, and I would see it as a global abstract, and then I would come up close, and I could look at the detail, and I was loving it. And then I started to let the metaphors flow, and I started thinking about this, and I thought, isn't this just like real life? That we're measured by and reviewed by others by the company that we keep, and don't we have an effect on that group? And then I started thinking about the accident of birth and chance in life, and I was really on a roll, and I was really excited about it. And then I exhibited with really a lot of great instructional text, and during the show, people gathered around me, and they said, what the hell are you talking about? And so anyhow, I'll show you what the hell I'm talking about. And so they would change, and I would vary the speed. And if I slowed it down like this, people loved it. If I speeded it up, they complained because they didn't like having to not focus on the faces. And I'm not exactly sure where this is all going, but I was having fun with it. But it's part of the process. It's how one thing leads to another. You experiment, you see something visual, you play with it, you say, aha, I've got something. Some ahas aren't ahas, some are ahas, and it's great. So here's another real quick one. All right, back to Rodin. So I'm at the Met, and I'm standing in front of, those of you who know, the Burgers of Calais, and I'm going, oh, this is so great. And I walk around it, and I say, it's so great, and I walk around it some more. This is so great. And it occurred to me that it's so great at an infinite number of viewing angles. And I've only got one as a painter. It's right there, it's 2 dimensions. And I thought, now how can I do this? Well, computers, of course. And so I bought a program that would help me give the perception of actual depth and actual volume and give me an infinite number of viewing angles. So I had this great mannequin that I was able to form to the face I wanted. And then I was able to take my drawing and paste her on it. Now, don't get sick. But I can do all these great things with her. It's going a little slower and I want it to go, but it's back to The Exorcist. I've got her walking, I've got her doing all these things, but I'm creating this 3-dimensional drawing, and it's great fun. I'm not there yet, but it's how I work. It's the process. But I love this notion of sculptural-like depth and volume. I love the idea of violating the sacrosanct flatness of modernism and just playing with something new. Okay, let's wrap this up. So I briefly talked about a bunch of subjects, but there's a common thread there, and it's about process and discovery. Experiments, mistakes, accidents, and observation. It seems pretty basic, and there's nothing new. Artists have been doing it for years and years and years, but it was new to me. And today, while I'm still trying to understand how these visual perceptions translate to an emotional response, at the very core, I blew that, but okay, I'm going to start this paragraph over. And while, and now, and while I'm beginning to understand the ways that people are visually drawn to certain aspects of the physical art itself, that being faces, eyes, line, color, and light, at the very core, I do not understand why some art elicits an aesthetic or emotional response. But I just see that it does. And so here's just a very fitting quote to end on, Francis Bacon. Thank you.
Panelists to come up here. This is perfect. We have the artist ask a question, and as we know, science can answer every question. So in just a minute when they get a seat, we will have a definitive answer on how paintings create emotional responses. Actually, this is the time when it's time for you to get involved here. So I think we have a couple of people wandering around with microphones. If you raise your hand, they'll come. There's one over there. They'll come around. So while you're getting ready to ask some questions, please put your hand up if you want a mic to come to you. But I have a question or two. I thought we might just start off with it. I will not ask you the deeper question of how what we see creates these emotional responses. But I thought maybe we could start with just one aspect of Bill's work, and that is he works with faces and with red. So scientists, help me out. What is that doing to us? Marge, you want to go first?
Well, I work on low-level vision, and Jeremy works on even lower level.
Feedback.
And Jeremy works on even lower level vision. So we're not exactly experts. We're not exactly experts on answering that sort of high-level question. But I will say that there are large parts of our brains dedicated to processing faces. It's actually my opinion as a scientist that is because we spend a lot of time during development seeing faces and knowing that faces are important for us. And so I think that's also true of red. I don't think there's anything privileged about red, the red pathway, except that we learn culturally and experientially, wow, that hurt, that red is a hot color. It's an emotional color.
And Jeremy, I was intrigued. You talked about how the world looks to, say, honeybees, and you know, as we all know in evolution, many things are conserved from honeybees up through people. Is there something that may be conserved from very early part of evolution that may be triggering something in us when we look at some painting that Bill has done?
My guess is that it's specific to our species, and even specific to our species now. I mean, my guess, and this is more the naive observer than the scientist speaking, that when I see his paintings, there are two separate channels, two separate ways in which I'm taking in the painting. One is the more analytic, looking at the face, sort of straightforward, viewing the world channel, and then the simultaneously and sort of subconsciously, the red paint is adding an emotional overlay on top of that. And that, quote, colors the way I see the face.
What do you think, Bill?
Well, I've given this a lot of thought, and I can tell you, I don't know. I like red.
This is good. We'll start off with not knowing, because as we mentioned before, a lot of this conference is about what we don't know, and it's about questions more than it is about answers. So let's have some questions. Who's got a question over here?
As you beautifully showed, Dr. Nathan, despite our strong perception to the contrary, the visual world is deeply and inherently ambiguous. So what does that say about how the brain forms our perceptions?
Well, I think, maybe I should repeat the question. So the point was that there's an ambiguity inherent in the way we're processing visual information. Actually, Samir Zeki has thought about this very deeply, and especially in its relationship to art. I think we are, in a sense, sculpting the image, the meaning of the image, especially from our own experience. I mean, the low-level examples I showed, for instance, the subconscious or unconscious assumption that illumination is from the top, that's experience. If the sun were down below, if somehow the world was illuminated by a light source beneath our feet, we would see the cuneiform in reverse. I think that effect permeates our perception. I mean, everything that we're judging is based on those kinds of experiences, conscious or subconscious. Samir, what do you think?
Could we get a microphone to Dr. Zeki? Over here.
So maybe you should come to the podium.
I think we got a microphone is on the way here. Dr. Zeki.
Thank you very much. Look, I've got a number of questions and comments, and of course, there's going to be some disagreement. It would not be a scientific meeting if there were any disagreements. First of all, on the question of ambiguity, I think color is one area where there is no ambiguity. And I think this is due to the fact that it is an inherited concept. I mean, a green is a green is a green, whatever happens. And you cannot easily play around with that. But I want to come back to that. First of all, let me just attack Marge here, because she said that she did not study higher functions, but you do, because you study faces. And the question I would like to address to you is, do you think that faces, you made the statement, which I'm not sure I agree with, that we spend a lot of time, a lot of space given to the perception of face in the cortex because we spend a lot of time looking at faces, but I would suggest that faces are an inherited concept. That
I know that most people think that.
And why do you not think that?
Well, so my lab did a lot of research on faces, and I realized as we were doing this that there is this idea that's prevalent among a lot of people that face recognition is innate because of the biological importance of it. But we also have, in almost exactly the same part of our temporal lobe, a part of our brain that's for processing letters and words. And that's not innate, right?
No.
So we started asking, and so I thought, well, there's this discrepancy between the innateness of faces and the clearly unnaturalness of reading. And so we started teaching very young animals to recognize symbols. And clearly that's not going to be innate, and it turns out they developed an area too for recognizing symbols. So I'm beginning to think that intensive early experience can cause modular organization in the temporal lobe in the same way it does in early sensory areas by experience-dependent Hebbian mechanisms. And you've got so much experience with faces from day one.
Well, I'll tell you.
So that's an easy mechanism, and it's already got a history for that.
I'll tell you of another experiment in which you show people deformed faces over a long period.
Adults or kids?
Adults. Hang on. Don't dismiss it. And the activity in the frontal cortex does not diminish because they never adapt to it. Any departure, that's quite interesting from the point of view of Francis Bacon's really deeply deformed faces. So I'm not sure. In any case, children recognize, newborns recognize faces very, very quickly. So I suspect that's more of an innate.
I am aware of this bias in the field, and I am doing research now to ask if that's really the case, and I'm becoming skeptical.
Okay. In your cells, the cells that respond to faces, you said some have respond to some inter-eye distance, specific inter-eye distances, others to narrow ones. Are there any ones which would respond if the two eyes are outside the face?
So that stimulus we made, we got the eyes as actually out to the very edge of the face. Totally implausible.
Yes.
And that was where you got the peak response. I don't know if it would keep going if the eyes were out here. I have no... we went way outside plausible faces, and yet we still got ramp-shaped tuning.
So
We were
you did a
surprised.
Francis Bacon. Then.
Yes, exactly.
Well, I don't want to hog the whole thing. Maybe others can ask, and then I'll come back because I've got plenty more questions.
I'm sure you do. I'm curious, Bill, did you want to add, I was curious, you talk so much about eyes and not just faces.
Right.
And so since we got into this thing of when you move eyes around and stuff, how do your eyes, you say you don't use the regular colors, but otherwise are they represented as what we consider the kind of eyes we see?
Yes.
And you think that's an important connection to people?
They respond to it. And, you know, sometimes, actually, the image that you showed, the eyes are kind of askew. And it's the same thing. But I haven't experimented with taking the eyes outside of the body or making them Picasso-esque. I haven't done that yet. Maybe on the docket.
Right? Next
I
time you're
love
here?
the way you get motion in your eyes by using equal value.
Yeah.
That's
and that was
phenomenal.
pure accident.
Or pure science, if you look at how color and motion are tied together. We have a question over here.
Hi. My wife, when she is asking for a kiss, well, my wife wears lipstick, and she wears red lipstick. And just getting back to your the idea of red, and it seems to be the most carnal color, and you're dealing with heads, rather beautifully done heads, and using red in its most carnal fashion. I would be very interested in knowing how you might approach the same heads with colors that are less carnal, less appealing,
Mm-hmm.
because you're using red in a very accessible way.
Yes.
And so it would be interesting to see you move someplace different. And again, I'm also thinking about babies' cribs and having that little smiley face that we kind of prop up above them. We tend to kind of always look at eyes, and so it's a nice way for people to spend time with work is always to go right there. But it would be interesting to see you move into another direction.
Yeah, and actually I've done that.
And Bill, before you start,
Sure.
maybe you could also talk just a little bit about how you perceive red. You mentioned having some color vision impairment.
Mm-hmm. I see red as red. I think I do. At least I can differentiate red. But I always treat red like black. And so I will interchange them sometimes. I can't tell you why. I just started using the red because I like red. It's not blood. Well, that's what a lot of people say. Oh, you put blood all over them. That's really not my intent. If you choose to see it that way, that's perfectly okay, and I love that. But it's not where I'm going with that. I told you I like to use Photoshop. I'm always, because I'll go, man, I've been using red for 2 years now. Let's try something new. I pop it into Photoshop and I'll make the red green or blue or purple. It doesn't work for me. Now, the only color I've found that works for me in this same way that I'm either putting it on the face or putting it on, it appears like a screen in front of them, like it's a separate abstract expressionist painting, is orange. And again, it's so similar to red, and it's a kind of a reddish orange. And I started doing that on the bigger paintings. I hurt my right arm. I couldn't use it for 4 months, and so I started drawing and painting with my left arm. And all the big pieces there, like in the beginning that had the orange on them, those are all left-handed pieces. And it surprised me that I was able to do this. And I think it also got into, I went bigger because I couldn't draw smaller, and I think that had to do with my motor skills, small motor skill, as opposed to this. But just for the heck of it, I changed to orange for all the big paintings. I kind of said, well, I'll make a break here. I'm painting left-handed, and I'll do these orange. And the orange works. But they're definitely different because the orange pops out and the red hangs back. I won't say it recedes, but it's kind of on the same plane. And so now if I want the abstract expressionist marks to look like they're flying off at you, I'll put them in orange. If I want them to be more part of the picture, I put them in red.
And do you think that the reds that you see in your paintings are as intense to you as they are to people with typical color vision?
Probably not. That's what I think frequently. Okay, so I take a Ishihara test, is that right? Sometimes I I can't even get half of them. I wanted to be a pilot, and that's critical, being able to see colors. And then other times I'll do a little better. So I think it's lighting, it's all kinds of things. But that's the red I've always seen, so it's hard for me to say whether someone sees it stronger or less so.
Sorry?
Oh, it's, okay, you go to the eye doctor and they have these dots, you know, and there's a number or something in there, and I always give them the wrong number or I can't see it at all.
So clinically demonstrated color blindness.
Oh, yeah.
Okay. Got it, got it.
Color anomaly.
Impairness.
Color impairedness. Fair enough. Yes?
Hi, I'd like to offer an example in architecture of color use that is counterintuitive and get your comment on it. We designed a school a few years ago for highly distractible high school students who were not risk averse. They were the opposite, whatever that term might be. And it was a 4-story school building, and so they had to use the central stairs extensively. And what we did was paint those stairs with highly stimulating colors, lots of contrast, and when they got to the hallways, the long hallways that connect to the classrooms, we moved down a step to lesser contrast, less saturated color, and when they entered the classroom, extremely mild color and no value, very mild value. And the teachers report that the students were able to calm down through that process by transitioning between spaces. The idea, initially, the school reacted very badly to the idea of painting saturated colors in the hallways. So I'd like to get your response to that. And I'd also like to mention a book that might be useful called Survival of the Prettiest by Nancy Edkoff, where she summarizes a lot of experiments about faces, the average face, babies' reactions to pictures of faces, and how those reactions seem to match those of adults. So if you could give me a little help on the color in the stairwells, that would be of interest to me.
Who wants to go first? Jeremy, take a shot.
It's different from my high school.
And how was your behavior in high school?
We better not go there. That's very interesting. I think that's the sort of thing you could imagine in extreme form, you know, for example, in the military. I mean, why do people all dress the same in the military? Well, there's a huge effect on behavior of how you view the world around you, yourself. It sounds highly plausible, what you're describing. Sort of subconscious, but plausible in terms of its effect.
I think it was time for one more question. How about right here?
Hi, I don't know the microphone's on now. Speak loudly.
Recording
Can you,
this.
I just wonder if you can maybe
Excuse
step
me.
back a little bit next
We're
to the
recording
dots.
this. Can you give her a mic?
Hello, is this better?
Thank you.
Yes. I'm wondering if you can maybe step back a little and connect some of the dots. I understand that, you know, color provides all kinds of useful survival information. What would be the survival advantage of loving, you know, an art on a canvas, you know, something by Bill or something by Monet?
I think as a biologist, I would say, rather unkindly, it's an epiphenomenon. It's simply because our brains are so powerful and have been designed for so complex a series of tasks that art has basically fallen out as a side effect. Now, of course, it's one of the greatest things that could possibly have happened, but I don't think it was part of the Darwinian selective pressure that got us where we are today.
Well,
But much of
unless
culture
you
is that
count
way.
the ability to communicate.
Marge, you want to say a little bit more about that?
Well, I think art comes out of communication, which is, I think, a fairly low-level survival skill. Artists are communicating with other people.
Bill?
Well, you know, that's what I hope I'm doing. And so I go back and forth, and I will say, okay, Guernica communicated something. Am I communicating something? And sometimes I think I am when I'm full of myself, and sometimes I say, no, I'm not, and I get frustrated. So I really don't know the answer, but the hope is that it does.
So I'm sorry, so Jeremy, so are you suggesting that it's, or saying, you know, that it's, you do not think it's, you know, that it's the artist, our love for artists and learned response and not a inherited response?
No, not so much that it's in terms of learned versus inherited, just that we're not hardwired to be appreciators of art at a sort of Darwinian level, we simply have evolved an enormously complex central nervous system out of which art appreciation has naturally fallen. But that wasn't the thing that, you know, found the mate or got the next meal.
I mean, the reason I'm understanding is out of which it was naturally fallen.
Let's see, how to explain this. Yeah, the question, so what does one mean by, you know, how did it just sort of fall out?
And by it, go ahead and repeat the whole question.
Yeah, so the question is, you know, how could you imagine a system getting complex and then properties emerging from that complexity that you wouldn't perhaps have expected or weren't initially built into it? I think there are many, I mean, I don't have sort of an answer, but I think there are many examples out there of that sort of thing. If you look, for example, at social complexity, say the economy, you know, the stock market, for example, there are a series of individual players, each trying to make money, and yet the crash of 2008 was not predictable from each of those, maybe it was, but not by me, each of those individual elements. I think there are many examples when you get a level of complexity that's extremely high, you get a series of emergent properties that were not simply predictable from the individual pieces. And I would say art appreciation is in that category.
On the other hand, we often agree on works of art that are good. So it's got to be a mixture of cultural evolution and tapping into low-level things that we all share.
Aesthetic disagreements. We're going to get into that more in later sessions, but we're about out of time for this one. So I'm going to thank all of our panelists for speaking today. Thank you. There'll be about a 15-minute break, and then we will reconvene to listen to some music.